My project studies astrocytes and how they impact plasticity in the developing cortex with the aim of understanding how learning rules are modulated in healthy brains to help us study disease states. I'm going to present the first steps that we've taken in our lab using electrophysiology and 2-photon microscopy to characterize astrocytes in the developing visual cortex and to look at the impact of a gliotransmitter, D-serin, on cortical plasticity in acute brain slices of mice. Astrocytes are a type of glial cell which have processes that associate around neuronal pre- and postsynaptic elements to form a tripartite synapse. Here, they are in a strategic position to bidirectionally communicate with neurons. Astrocytes possess numerous receptors for neurotransmitters and respond to synaptic activity by generating intracellular calcium activity. These calcium transients can trigger the release of neuroactive signaling molecules called gliotransmitters, which include glutamate, ATP, and D-serin. Gliotransmitter release enables astrocytes to signal back to neurons and has been shown to modulate neuronal excitability, synaptic transmission, and plasticity. Oftentimes, alterations in astrocyte function precede neuronal abnormalities in early stages of diseases, such as epilepsy and Alzheimer's disease. Therefore, understanding how astrocytes modulate neuronal function in healthy learning and memory is an important first step. To characterize layer 5 astrocytes in the visual cortex using electrophysiology and 2-photon imaging, we used a synthetic fluorescent dye called SR101, which is selectively uptaken by astrocytes. This allows us to use 2-photon laser light to excite the dye and visualize the cells for targeted patching. Here is an example 3D stack of a patch astrocyte with a dye-filled pipette showing its highly branched and bushy morphology. Based on our preliminary data, we found that the electrophysiological properties of layer 5 astrocytes were comparable to those reported in literature. They are more hyperpolarized compared to neurons and have low input resistance. In addition, there are potentially diverse subtypes of astrocytes based on their membrane conductivity, which we found after stepping different voltages across the astrocytes. Some cells, like the first example, showed typical passive conductance, whereas others, as in example 2, had a time-dependent slow activating component at larger voltages. Astrocytes are also known to communicate with other astrocytes in their network through connections via gap junctions. So we quantified the time taken for half maximum dye filling across connected astrocytes as a function of gap junction coupling. We found through clustering analysis that astrocytes connected to the patch cell can be grouped into primary coupled cell and secondary coupled cells, which correlate with distance away from the patch cell. We hypothesized that the primary coupled cells are those directly connected to the patch astrocyte, and the secondary coupled ones are those connected to the primary cells. Furthermore, loading the fluorescent calcium indicator fluor5F enabled us to see that calcium transients occur spontaneously in waves. By identifying different regions of interest, we can see that these transients start from one branch of an astrocyte and spreads to other branches. Our other study looked at the impact of a gliotransmitter D-serin on plasticity. Spontaneous neurotransmission is synaptic release independent of presynaptic firing. To investigate spontaneous release, we performed whole cell recordings of layer 5 pyramidal cells, which are cells that mediate the majority of outputs from the neocortex. And we recorded miniature excitatory postsynaptic currents, or EPSCs, in the presence of a sodium channel blocker, TTX, and a GABA-A antagonist, bicuculin, before, during, and after D-serin wash-in. We also looked at evoked neurotransmission, which occurs following a presynaptic action potential, because our lab and others have shown that spontaneous and evoked release are regulated through different pathways, so the responses are expected to possibly be different as well. We use quadruple whole cell recordings to identify monosynaptically connected pairs where excitatory postsynaptic potentials could be elicited in a postsynaptic cell by spiking the presynaptic cell. We found no changes in the frequency or amplitude of the mini EPSCs of spontaneous release with D-serin application, but we found downregulation of EPSP amplitude in evoked release. To summarize, we have laid the groundwork to investigate how astrocyte signaling correlate with short and long-term plasticity. We hope to further build on the techniques that we have implemented in the lab to study how astrocytes impact plasticity and modulate learning rules in the developing cortex, and how dysfunctional astrocyte signaling can give rise to neurological diseases. Finally, I would like to acknowledge my funding sources as well as my supervisor and lab members who have helped me in this project. Thank you for listening, and please feel free to leave a question if you have any.